exam to boost your confidence.
Oh.
Usually I give an easy exam for the first.
So nice of you.
Then it will slowly deteriorate. Okay, I want to first go over and give you a slide so you know what to remember, what not to remember. Okay. Delete. Hold on. What? Wait. Oh, this is not. You're not going to remember. Oh, it's just delete. Yeah. For your benefit.
Oh, this is from.
I mean, your goal is to make the book difference.
Right.
When I was a student, I always do that. Okay. And we know the cell, you have two cells, eukaryotic cells and prokaryotic cells. We delete. And there's cytoplasm and the nucleus for the eukaryotic cells. We need to remember.
Are you deleting things we don't need to know?
You don't need to know. I just mention what's going on.
You're saying.
Okay? Two kinds of cells. And the plant cell is eukaryotic cell.
Eukaryotic.
Yeah.
And the cellulose dimension for animal, 5 to 100. There are different scaffolds to make the cells rigid and having a transport highway.
Will you be posting these slides?
Yeah, I will post. I will combine them together. You need to know this one. There are 4 most abundant elements in living organisms: H, O, N, C. Hydrogen, oxygen, nitrogen, and carbon. Okay, in dry week, carbon, nitrogen, oxygen, and hydrogen. You don't need to know why there's a carbon-based alive and a silicon-based alive. Okay, you need to understand what is the configuration and conformation. In a cis-trans isomer, in enantiomers or stereoisomers that are mirror image of each other, when you rotate, they don't superimpose with each other. Remember this? And living organisms exist in a dynamic steady state, never at equilibrium with their surroundings.
Cool.
Second law of thermodynamics, the entropy keep increasing. Means the randomness keep increasing. Oops, sorry. I shouldn't do it. And gives free energy, dirt G equals to dirt H minus T dirt S. Understand the terms. And know the difference between exergonic and endergonic. Dirt G less than 0 is exergonic. Dirt G greater than 0 is endergonic. You should know which one is a spontaneous reaction. When dirt G less than 0, that's a spontaneous reaction. Understand the difference between the two, and know how to calculate delta G from delta G0, and we know the concentration of products and the reactions at the equilibrium. We know the K equilibrium, so RT law and K equilibrium plus delta G0 equals to delta G. practice on this one. So the difference between catabolism and anabolism. The catabolism means you create free energy. And anabolism means you require the input of energy to produce higher order molecules. For now, you don't need to know DNA, because we're going to talk about that in chapter 8.4. So evolution. Okay, that's for chapter 1. We have 17 lines left.
Thank you.
Chapter 2, there will be, majority of the questions will be pH questions, PI questions, and what is the hydrogen bond? I think rough idea of the bond energy for hydrogen bond and covalent bond. It's about 20 times as we compare a covalent bond to the hydrogen. when pH, note that at pH equals to pKa, the concentration of the conjugate base equals to the concentration of the weak acid. So when you further titrate, when you add more strong base to the solution, titrate to the right, you will have more of the conjugate base.
Okay.
Yeah.
The buffer zone, is that like a, it said in the book it was, plus or minus 1 pH. Is that a given, or is that like a general thing?
That's a general. I'm going to give you an example of, there will be a question about the weak acid with more than one dissociation, proton dissociation, more than one ionizable groups. So you need to know at a particular pH, what is the dominant species? For example, phosphoric acid. Like this. And you should practice on this one. There will definitely be a short answer question like this, but instead we will add either a strong base and shift the equilibrium to the right. Remember, if you add a strong base, shift the equilibrium to the right. Add a strong acid, shift to the left. I give you the final pH, you need to calculate the pKa value, or give you the pKa value initial pH, you calculate the final pH, then add strong base or strong acid.
I'm assuming
it will be very similar to such a question.
It won't be
change some numbers.
It won't be this easy to where we can guess it conceptually, though, right? Like this one.
It will be this easy.
Oh well. If we do it conceptually, though, we won't. Our work will be. We're just going to justify why it would be A or whatever. I mean, if you do it conceptually, we wouldn't have to. Do you need us to show an equation of why we?
Yeah, so you need to show.
So we can't do it conceptually. We've got to show work. Okay.
Yeah.
Okay.
It's not a. There will be one short answer question.
Okay.
Like this.
Okay.
You need to understand the buffer concentration equals to the weak acid plus the conjugate base, and then combine with the Henderson-Hessenbach equation, you solve the unknowns. Practice on this question. And try to, I suggest you try to do the calculation using each row of this table. And find out what's the change in pH. Okay, that's for chapter 2. Any questions for chapter 2? For the amino acid, there are 20 amino acids. I will give you the structure of all 20 amino acids.
Oh, that's nice.
You need to understand the different category. We have a non-polar aliphatic amino acids, 7, and 5 polar and charged.
Do you want us to memorize the shorthand? Like, are you going to give us like a chain shorthand notation?
I'll give you the name of the amino acid and the structure of the amino acid.
I mean, like the 3 letters.
3 letters.
Like you said, we're going to have one that's like that non-peptide problem.
You probably don't need to know the 1 letter for the exam.
Cool.
You will have enough information for it.
Okay.
And we have 3 aromatic amino acids and 2 negative charge, 3 positive charge. Pay particular attention to the histidine. Talk about this.
Being so cryptic.
What was that just? Well, because it could be 1 or 0.
There is know the term of the Zwitter ion at the physiological pH. The alpha amino group will be positive and the alpha carboxyl group will be negative. And understand you will have questions like I ask you at pH 1 for this amino acid, like histidine or aspartic acid, and tell me the net charge for that amino acid. And if we increase the pH to 3, 5, 7, 9, 10, 13, 11, 13, what is the net charge for that amino acid? If pH equals to, for example, pK1, and you should know the ratio between the conjugate base and the weak acid. So roughly know that the pK1 for our carboxylic group, alpha carboxylic group is about 2, and the pK2 for the alpha amino group is about 9. So at pH 1, the alpha carboxyl group will be
Hydrogenated.
which form? Neutral or negative or positive?
Neutral.
Will be neutral. It's protonated. Then it will dissociate a proton. Then when we reach to close to 9, the amino group will dissociate the proton. Okay. Use these examples to go over the dissociation of a proton from side group. For example, aspartic acid and glutamic acid. And lysine, arginine, histidine. Note that the histidine, the protonation of the on the fine membrane, the position of protonation. Okay. Then there are two kinds of amino acids that contains the
Methionine,
thiol
cysteine.
group, methionine, and cysteine.
Mhm.
I will give you the table like this, so you will know the pK1, pK2, and pKr. Understand what is the PI value and the effect of the PI and related to the pH. If the pH is less than the PI value, what is the charge of that peptide or protein?
Negative. Positive.
It's going to be protonated,
Protonated will be
So-
positive. When we increase the pH, then it will be
deprotonated.
deprotonated, will be negative. That will affect the, when we use the ionic exchange column to separate the protein, that will affect which one will come out first. If it's a cationic exchange,
Then
which one will stay on to the stationary phase longer?
Protonated.
Cationic, what is the cationic exchange?
Oh, is it
Negative
negative?
Negative bead,
Right.
and you run the protein through the positive charge, the protein like to stick to the.
Catches cation.
Yeah, that's
Cool.
cationic. And the PI value, the PI lies midway between the two pKa values that indicate the protonation and deprotonation of the electrically neutral form. If it's a glycine, it's just half of the pK1 and pK2. If you have a R group, then you need to look at the neutral form. You find out where is the neutral form, and then you add up the two pK values around that neutral form, and then divide it by 2. That will be your PI value of that amino acid. So let's say if I give you such a graph, and I point to a different pH, and then you need to be able to give me which species exist at such pH. that clear?
To the major speed.
No? So you need to look at, I recommend that you carefully, very carefully, look at this 1, 2, 3 graphs. Okay? For those of you who are not here, you don't need to tell them. Peptide bond, what is a peptide bond? It's between the carboxyl group, alpha carboxyl group, and the alpha amino group. They come together and get rid of one water, right?
Mm-hmm. Condensation.
It's a condensation reaction. Okay, there might be some question. In your homework, you have questions like this. There's a polypeptide chain with a few amino acid residues. At a different pH, what is the charge of that peptide, right? Mm-hmm. And when you use the ionic exchange column to separate the peptide, which one will come out first? Then it also asks you about the molecular weight. If there's a larger molecular weight peptide and a smaller one, which one will come out first from the size exclusion? Mm-hmm. You will see such a problem. The problem with the exam is that we only have 50 minutes, so we cannot really write too many questions. Mm-hmm. So you can see we already have a lot of material for you. So you need to understand the principles of the column chromatography, ionic exchange, so separate the proteins with different charges. In size exclusion, the larger protein will come out first, the smaller one will come out later. In affinity chromatography, the isoelectric focusing, what's the principle of isoelectric focusing? To create a gradient of a pH in your gel in a column. Then when you run your protein sample, and depending on the PI value, if a protein has a certain PI value, it likes to go to that pH gradient. And cysteine will form disulfide bonds between cysteine and cysteine. And send how to sequence the polypeptide. There will be a short answer question similar to the class problem we had last lecture. Even simpler. Yeah, the one,
So it'll be like that one where you provide the agents, we need to know the agents?
Provide the information, okay, you need to know what is the N-terminal, and which chemical we use to identify the N-terminal? FDNB, right?
And are you going to give us that little table of the...
I will give you the table.
Right, the enzymatic.
Make sure you remember.
Well, it's important. It's important. We either have to memorize it or
the mechanism of need to know what is going on here. Okay, this is important. I will apply this table in the exam. And remember, trypsin will cleave at the C side of lysine or arginine, and chymotrypsin will cleave on the C side of the three aromatic amino acids. What are the three?
Trypsin, tyrosine, and phenylalanine.
No, trypsin. Tryptophan.
Oh, tryptophan. Thanks. Tyrosine, tryptophan.
Tyrosine,
phenylalanine.
Pepsin will cleave on the inside of the three aromatic amino acids plus
leucine.
a leucine. Cyanogen bromide on the C side of methionine. Okay. So this question is harder than the exam question. Okay, that's chapter 3. Any questions for the review, for the content? Material that will be covered in the exam. You have questions for the homeworks?
Do we use
Class problem?
Yeah.
Scantron?
Yeah, you use the Scantron. Scantron and short answer you need to fill in on paper.
Can we bring our own again? Can we bring our own Scantron?
We will have those slides.
Oh.
Chapter 3, total 22, they told you that the FDMB identified alanine and leucine as amino terminal.
Right.
That means you have two terminals.
Two terminals.
Right? Two terminals means what?
Two chains.
Two chains in that subunit in a protein. Okay, contains two copies. One copy of each chain, but two chains. Okay, doesn't mean you have only one copy. Okay. From B, you have 80,000 molecular weight, and then after you use a performic acid, what does performic acid do?
Hydrolyzes.
Break the disulfide bond, right? And then you use the gel. You identify there is a molecular weight of 35,000 and 45,000. That means the disulfide bond break it was broken by the performic acid. Okay?
Each one of those parts?
Yeah, so
each chain,
gets into two parts. C, size exclusion chromatography indicate the native protein has an apparent molecular weight of 160,000. It doubled the 80,000 using a denaturing gel, the page gel, SDS page gel. Okay, that means this protein is a homodimer, has two copies of the subunit. Each subunit contains two chains. Homodimer means this two is a twin copy of the two domains of protein. They come together.
Two dimers?
Yeah, it's a dimer. Okay, that's the information. You have one subunit and another subunit.
Okay, all right.
Each subunit has two chains.
Okay.
Same sequence, okay? And connected by thiol group. Maybe more than one thiol, more than one disulfide bond. Okay? So when you run onto a SDS page gel, only get denatured, so you see only one copy. All right? However, when you use the disulfide bonds, you break this, you have two chains. And also because of the two terminal alanine and lysine, whatever you identify there are two terminal, the two chains. Okay? But when you have a size exclusion, it's a native form of that protein. And you measure the molecular weight is it doubled.
160.
So you know there are two copies of the same protein.
So since that's made
subunit or subdomain.
Since it's made of 4 polypeptides, do we call that a dimer or a tetramer?
we call a dimer of the
Dimers.
subunit. Okay, the dimeric protein, homodimer,
And each subunit is a dimer.
containing 4 polypeptide chains.
So what's the difference between that and
A tetrameric
before, though, right?
protein means you have 4 domains, 4 subunits. So this might have contain like each may contain like 3 chains or 5 chains. We don't really use how many chains you have to define the dimer, trimer, or tetramer. Okay. Oh, this one.
What number is it?
15. 3-5.
We went over this in the class. It's exactly the same concept. Okay, now this. So here homework number 2, number 15. The compound is going to have a free amino group with a pKa of 8.8 and another ionized group with a pKa between 5 and 7. All right, there will be a question like this in the exam. So you have A plus inserted into A0. You have 2 ionized groups. That means you have 2 pKa and A minus. And pKa1 is between 5 and 7. And pKa2 is 8.8. Okay? And the initial concentration we know as C0 is 0.2 molar. It's 100 mil. And the initial pH equals to 8.2. Right. Then we added 14 mil of 0.2 molar HCl to the solution. Okay? And the pH got changed to 6.2. And the question is asking for the pKa1. What is the pKa1? So when you see the initial pH equals to 8.2, this is closer to this pKa2. So we're going to use this part of the equation first, right? Then we need to identify the concentration of the conjugated base for this part of the equilibrium and the weak acid. We know the pH equals to
Can you switch to black?
black.
Yeah, if you don't mind.
Worse.
Oh, is it? Oh, it's worse. Oh. It's all good. Huh? Oh, there's it. I'll buy some black.
pH equals to pKa2, right? Plus the conjugate base is A minus divided by A0. Okay? And we know the pH is 6.2, 8.2. pKa2 is 8.8. Okay, so just do the subtraction. Minus 0.6. Okay, the anti-log is
4.
6.6.
4.
1/4, right?
No, it's like 4.
No, 4. Right.
1/4.
Wait, what is it, minus 6?
Oh, sorry.
Minus 0.6. 8.2 minus 8.8.
Oh, it's 4. Sorry. Yeah, he's right.
0.25, yeah.
Equals to A minus divided by A0. So A0 is 4 times A minus. Okay? A0 is 4 times of A minus. And we know A0 plus A minus equals to the total concentration of the buffer. So A0 plus A minus equals to 0.2 molar. Okay, so we plug in A0, that will be 5A minus equals to 0.02 molar. So A minus will be 0.2 divided by 5, that's 0.04 molar. Okay? Do a calculation of this. 0.04 molar. And this is, A0 is 4 times A minus, then this will be 0.16 molar. Okay? Follow you me?
And what's the, did you use molarity for that equal signs, or do we need moles?
Yeah, molarity. So A0 equals to 4A0. So if you substitute this one, let's see. 4A minus. Right?
Yeah.
That's 5A minus equals to 0.2 molar molarity. Then A minus equals to 0.04. And A0 equals to 4 times A minus. So this one is 0.16 molar. Okay. And then you times the volume. Initial volume is 0.1 liter. That's 0.1 liter. Okay? So this equals to 0.004 moles. This is 0.016 moles. This is the initial condition. Initial condition. Then we add 0.2 molar of 14 ml HCl. How many moles of HCl did we add? 0.2. You like the red.
Darn it, sorry, it's all good.
Yeah. Point 0.2 molar times 0.04, that is 0.008 moles. Okay? HCl. When we add this much HCl, it will react with the base, the conjugate base, and form the weak acid first. If we have an excess amount of HCl, it will drive this further to form the A plus. Yeah. Yeah. If you add a strong acid, it will drive the reaction to the left. If you add a strong base, it will drive the reaction to the right.
And we added an acid.
We're adding a strong acid. Yeah, it's going to the left. So let's say you have a point.
Why do you multiply by 0.04?
Volume.
Molarity times volume.
Yeah, this is 0.04 molar. We just calculated from this. Okay? And this is the how many liters we have. The volume. So molarity plus volume equals to moles. The quantity, how many molecules you have.
So why that and not the 0.016? Why that and not the 0.016? Because you multiply by 0.004. So
Oh, he just did that for the acid, not for the original weak acid, for the strong acid over here. He was standing in front of it.
Yeah.
oh yeah.
This is for the
That's
weak
right.
acid. This is the strong acid. How much is strong acid? Okay? So we know that this is 0.008, but this is only 0.004. So we have excess amount of the strong acid. Now how much A minus will be consumed, will react?
All of it.
Only 0.04. We only have this much, right? So this will become 0. All of the A minus will be consumed by part of the strong acid. And how much will be formed?
0.004.
A0? Okay. That will be 0.016 plus
Yeah.
0.004. Okay? This is the consuming half of the strong acid to reach this point. And this point, this is 0, this is 0.02, and this is 0, right? Yep.
So in this problem, I guess you cannot add a base.
You can add it.
You
You
could.
can, but you just,
And it will go to the other side.
so what if you,
Let's finish acid first. Now you still have 0.004 moles of strong acid left to drive this reaction to the left side. That means we will further consume, the nothing left here will consume A0 to become A plus. All right? So how much strong acid do we have left?
0.004.
Okay, so 0.004 will react with A0 and form 0.004 moles of A plus. So we need to subtract 0.004 from this amount. That will be
0.020.
0.02 minus 0.004. Okay, that is 0.016. How much is this?
0.004.
0.004. This will be the final amount in terms of moles of A plus, and A0 and A minus will still be 0. Okay, so now you see the reaction gets driven by adding the strong acid to this part, right?
Mm-hmm.
We're in this equilibrium. Then we use the equation again. Now this is Henderson-Heisenberg equation. We will have to use pKa1 plus the conjugate base for this equilibrium is A0. What about this? A plus. All right. Now we know the amount of A0, right?
Mm-hmm.
You know the amount of A plus. You will need to divide by the final volume, but the final volume are the same, so we can cancel them.
Mm-hmm.
Okay? The volume. Okay. So log this will be equal to 0.016 divided by 0.004.
Mm-hmm.
What is the number?
Log of 4 is 0.6?
4.
Correct.
Okay. 0.016 divided by 0.004 is 4. Got it? Okay. So log 4 plus pKa1 equals to the final pH. This is what we want to know, but we already know the final pH. 6.2. Final pH is 6.2. 6.2 equals to question mark plus log 4. This is 0.6. Okay. In the exam, I will provide you a few logs: log 3, log 4, logs 1, log 10. So the pKa1 equals to what? 5.6.
There's so many errors.
You divide by the volume right there?
Yeah.
Divide by the volume,
Yeah.
and what happens?
Divide by the volume, you just plug in the number.
Yeah.
A0 is 0.016 divided by the volume, and A plus is 0.004 divided by the volume. Cancel the volume. Okay. Then use this to get to 4. The volume will be 100 mil plus 40 mil. 0.14 liter.
And what was it?
Yeah.
You said it was 0.004.